we're diving into a critical area, testing, validation, and verification. Get ready for an insightful discussion that underpins the reliability of food supply chain. The session will be moderated by none other than Dr. Lalita R. Gowda, retired Chief Scientist, CSIR, Central Food Technological Research Institute, Mysore, Member Scientific Committee, FSSAI, India Chair Scientific Panel on GM Foods, FSSAI. She needs no introduction. Most of us present here know her immense contribution in the years she has put in the food industry. Joining her will be Dr. Gyanendra Gongal, Senior Public Health Officer at WHO CRO, focusing on zoonosis, food safety, and One Health under the World Health Emergency Program. With 25 years of experience, he has led regional strategies, intersectoral collaborations, and published extensively on emerging infectious diseases and antimicrobial resistance. Next, we'll have Mr. Srinivas Rao, Head Quality Assurance, Hatson Agro Product Limited. He is a dairy tech graduate with 28 years of experience in dairy industry. He oversees ice cream, dairy ingredients, fresh and cattle feed divisions. He has also led key quality and production roles at Heritage Foods and Parag Milk Foods. Joining him will be having Dr. Edward Danish A., Chief R&D Officer, Fresh to Home Foods Private Limited. He leads the QA and supply chain integration initiatives. With over 25 years of global experience, he has in food aquaculture, food safety, and sustainable sourcing. He also specializes in FSMS and HACCP implementation. Ladies and gentlemen, please welcome all our panelists with a round of applause. I'd like to invite all the speakers to the stage.
Good afternoon, everyone, and welcome to this session. I think after the overwhelming lecture of Karthikeyan, I hope you have time to think this, because he put in so much of energy into this session, which I don't think would have allowed you to close your eyes. Although the session says testing and validation of this, that is a misnomer. We thought today we would discuss importance of antibiotic residues in the food chain, because this is a concern now as a safety factor in the food chain. So what I will do is just sort of give you an introduction to the antibiotics in the food chain, and my colleagues here on the stage, Dr. Gongal, Mr. Srinivas Rao, and Mr. Edward Danish will tell us how they look at it. Dr. Gongal will tell us about antimicrobial resistance, which again is a cause of concern globally, and the other two will tell us how they implement you're looking at the antibiotic residues or sourcing responsibility to see that we meet the regulations. So just let me tell you, to give you an introduction on antibiotics which are used in the food chain. Just I think to tell you that the discovery of antibiotics was by serendipity. So it was an accidental discovery, because when Alexander Fleming, he actually was working on microbiology, and he put his plate of Staph aureus, and he went away for the weekend, and forgot to put it into the incubator. But when he came back, what he saw is that there was a fungal growth on it. But unlike other scientists who think, okay, this is a negative result, by not throwing it away, he looked at it and he saw that there was a zone of clearance which clearly shows that this fungus was producing something which prevented the growth of this microorganism. And this is how the first antibiotic was born. And this fungus was Penicillium notatum, and the antibiotic was penicillin. I just thought this should be like an interest to the audience. Okay, so if we look at antibiotics, it's not a single word or it's not a single compound. We have different types of antibiotics, and in the antibiotics, these antibiotics actually work in two ways. And I'm sure most of you or all of you at some time or the other must have taken an antibiotic. So one of the ways the antibiotic works is that it either kills the microorganism, which is known as bactericidal antibiotic, or it is one which prevents the growth of it, which is known as bacteriostatic or antibiotic. And so that is why any physician that you go to and who gives you an antibiotic will ask you to complete the dose, because the dose which is given to you is to kill the microorganism that is there, and some of them are for preventive. But what do? After 2 days, fever goes down or something, we'll say, okay, I'm not going to take the antibiotic. But there is science behind taking the full dose which is given to you for 5 days. Now, coming to animal food or feed, if we look at the food chain, we have a host of animals with food-producing animals. That means these are animals which give us food, and they are bred and raised for the food consumption, and just like eggs, meat, or milk. And we have, in India, most of it are the dairy cattle, the goat, sheep, or maybe the broiler chicken and the layer chicken. In this industry, if we look at India's place in the production of these animal food, we are the 5th largest meat producer. We are producing about 7 million tons, of which 54% is red meat. We are the world's largest milk producer. We are the 3rd largest egg producer after China and USA. We are the 4th largest chicken producer after China, Brazil, and USA. And among the meat, it is chicken which is the most, or the maximum, which is consumed in India. So when you look at this global statistics of India's role in the food produced by thing, then the next thing comes is how are we managing it over there? So in the industry, what has happened is, most of the animals which are used for food production many of them are not from our country. They come, their original breeds are from other country and they have adapted here. And just like humans, these food producing animals are susceptible to infections by bacteria, by virus, etc. So therefore, in the industry, there is a rampant use of veterinary drugs, or we also call them as the antibiotics. So the antibiotics are used not only to protect them against the virus or the bacteria, but they are also used to treat healthy animals so that they don't get infected or that. It is like a metaphylaxis program that it is used. And in addition, many food animal producers, they use it as a growth promoter. So these are some of the wrong uses of this. And so therefore, if you look globally and you look at Codex, we have these regulations for the antibiotic limits, or we call them as the residue limits for antibiotics in the food that is consumed by the human. If you look at some of the statistics, to produce 1 kilogram of chicken, you need to use 148 milligrams of antimicrobials. So if you look, the production of the animals, which escalated from 63,151 tons, it's going to go up to about 105 to 596 tons. And so you can see how the consumption or the use of the antibiotic also will use, if you can do the maths of converting 148 milligrams per kilogram into what we are going to produce, like 1,05,596. So the amount of antibiotic used is going to be enormous. And globally, if you look, India actually, we are placed 4th in the use of antimicrobes for the consumption of the food animals, that is animals which are producing there. And the antibiotics that are commonly used in the animal or the veterinary industry are, if you see, we have the topmost one is the beta-lactams, then you have tetracyclines, the fluoroquinones, aminoglycosides, where penicillin falls, and this antibiotic consumption is going to increase in the animals alone by 82%. And their use in chickens is going to increase or triple by the end of 230. So what is the concern? The concern is what effect does this, animals being fed or given the antibiotics, for whether it is prophylaxis or metaphylaxis, have on the human. So it is known that, it might, antibiotic residues might get into the food chain. At the end of it, it would be there in the meat, it would be there in milk, and in the shrimp and fish that is there. And it goes into our system. It can have an effect on the antibiotic-resistant bacteria. We can have the genes for antibiotic resistance which are there. Our gut microbiome will change. And it is well known that if you have a healthy gut, that means if your gut is healthy, you're going to be a healthy person. So if there is any change in the microbiome of the gut, we know that it is going to have an impact on this. So how does a country take care of the use of these antibiotics? We cannot do without antibiotics. They have to be used in the animal industry because, most of the animals are susceptible, including the honeybees. The honeybees also are susceptible to fungal infections, and there's a lot of fungicides which are being used in the, honey industry. So if you look at this, FSSAI has taken, right from Codex, we have taken the thing, and regulations have been prepared. So every country has got regulations for antibiotics, and we call them as, antibiotic residues, or we call them as veterinary drug residues, because they do not stay as the antibiotic, but as products or metabolites of the antibiotic. So in the food safety and standards, you will find the regulations on what are the residue levels for the antibiotics in different foods in the food safety and standards, contaminants, toxins, and residues regulation 2011. And under chapter 2, you will have contaminants, toxins, and residues. And in that, under 2.3.2, you have the antibiotic and other pharmacologically active substances which are listed over there. If you look at that regulation, the first one says it has a list of antimicrobials and other drugs which are not permitted at all. So there is this list in which, none of these antibiotics are permitted. And then we have tolerance limits which are specified for seafoods, honey, eggs, milk, meat, and poultry. How do we meet these regulations? Most of you are food business operators. We looked at the e-commerce, Q-commerce. I asked the question, how do they manage the safety? They have to be tested, especially, if they are marketing fish, meat, egg. We need to be sure that the antibiotic levels are meeting the regulations of the country FSSAI. So for this, we do have testing methods. And if you look at the type of testing we have, we can do screening. There are kits available, like rapid kits, which within 5 minutes you can tell whether a particular antibiotic, not the whole group of antibiotics, but whether a particular antibiotic is present or not. And we generally use the lateral flow assay, or we use the fluorescent polarization assay, or radioimmunoassay and ELISA. These are all screening which will give us a yes or no. Now, if you get a yes, then you need to go a higher level of testing with the high-end equipment such as the LC-MS/MS or the GC-MS. And currently, India has, in our regulations, we have notified laboratories, and those laboratories which are testing for antimicrobials or residue analysis have to all be accredited to ISO 17025. And being accredited to ISO 17025 doesn't mean that they're all doing the veterinary drug residue analysis or the microbial residue analysis. They should have the test in their scope of testing. Only then an FBO, any of you FBOs wants to do the testing, you should undertake the testing in the ISO 17000 accredited laboratory where the drug testing or for the particular antibiotic is there in the scope of testing. So with this, I will close. This was just to give you an overview of what antibiotics are, how they are used in the food industry, coming from the animals which are producing the food, and what the government or what FSSAI has done to see that the food is safe and it has levels of antibiotics or antibiotic residues which are safe for human consumption. So next, I would request our next speaker, who would now tell you about what is antimicrobial resistance and how it affects the thing. So now I invite Dr. Jnanendra Gongal, the Senior Public Health Officer, World Health Organization, to tell us what he feels about antimicrobial resistance.
Thank you very much. First of all, I would like to thank organizer, Sarika Madam, and this Food Safety Works for giving me opportunity to share knowledge, expertise, and international dimension of antibiotic residue in foods. As Madam has mentioned, Alexander Fleming, he discovered antibiotics, that is penicillin, in 1928. And in 1929, he got Nobel Prize. And when he was receiving Nobel Prize, he mentioned that one day this antibiotic may not work because it will be available everywhere. It will be underused, overused, and abused. And that's why we are now dealing with this antimicrobial resistance problem. So, antimicrobial resistance, it is a biological phenomenon. It is applicable not only for antibiotics, it is for pesticide also, like DDT, malathion, we are hearing. So one day, insect or vector will develop resistance. So that is the natural phenomenon. And antibiotics are extensively used in human health as well as animal health, even plant health sector for treatment, prophylaxis, and growth promotion. And antibiotics means, antibios means it is a kind of biological weapon that that is for specific pathogen. But in our body system, there are bacteria which are helpful for our digestion. So when we have we take these antibiotics orally, it will also kill innocent bystander bacteria which are non-pathogen. And the use of antibiotics also promotes antibiotic resistance in non-pathogen. And these non-pathogen which are helping us in digestion, they also transfer that EMR gene to pathogen. So in that way, this antimicrobial resistance will develop, and more antibiotics, they are not useful. And we have to understand that in veterinary sector, in animal production, particularly in animal feed, poultry and pig, for pig production, we are using antibiotics. And first incidence of this colistin sulfate in pigs, it was found in China, which is considered medically important antibiotics. And there are also other studies. It is also used in poultry in Bangladesh, Indonesia, and many other countries. So research group, they already identify MCR1 gene. So that is related to colistin. So colistin is medically important antibiotics. And also vancomycin resistance strain also developed because of using this avoparcin in poultry feed. And therefore, the important message here is we should try to develop an effective system with surveillance for both antimicrobial use, that means in animal production, and antimicrobial consumption in human, and antimicrobial resistance. So what is the trend we have to develop? Antibiotics is extensively used in human health as well as animal health. So here, this is just example I am giving you that in self-medication. So even viral disease, we know that antibiotic will not work against virus. We have flu, but we'll take antibiotics. That is abuse. And if you have fever, you will go to counter medical pharmacy, you buy it, and once fever is over, then you stop. So there is no need of prescription. And that's why we have typhoid, recurrent typhoid, all those problems are there. And patient compliance, that is, I have already mentioned, and also over-the-counter sale. Even poultry farmer, they can go and they can buy it. They do not need veterinary prescription or any other things. And there are problem of counterfeit antibiotics in human health sector, but it is not a major issue. For veterinary side, the major issue is trade. So many poultry product or animal product which we export to European Union, it has been rejected because it contains nitrofuran or any other antibiotics residue. So this is the way we have to understand. What is the result of antimicrobial resistance in action by 2050? So initially it was thought 10 million people will die by 2050 if we do not take any action. That means our doctor will stand in front of patient helpless because the silver bullet is no more working. And now, according to new estimation, 39 million more lives will be lost because antibiotic is no more working. And 30% of them, it will be in South Asia. It is around 11.7 million. So that is the very critical issue we have to understand. And what is the health expense? Because antibiotic will not work, so hospital stay will increase, and people have to take not 3 days, 7 days, or 14 days. So in that way, 159 billion, it will be the expenditure. And global GDP reduction, it is 5.2 trillion, so that is affecting in poultry, animal production sector. And if we reduce antimicrobial use in animals by 30% globally, over 5 years, we could increase GDP 14 billion by 2050. It is according to World Bank statistics. AMR, definitely it is, as I mentioned, related to international trade, codex, OIE, now known as OHA, that is taking care of animal feed particularly, and animal welfare, and World Trade Organization. So importing countries or trade bloc, they impose standards and restriction. So that may include not only livestock and poultry products, but also fish, seafood, as well as honey. And what are the critical factors to consider when importing? So there is so-called performance of veterinary services. India has done in 2018, and it is a risk assessment. So they will try to see what is the regulatory framework, how it is enforced, whether there is a disease surveillance system in place, how effective is it, and whether there are laboratory capacity for investigation, detection, and validation of antimicrobial resistance. So these laboratories should be accredited, and there should be sample flow, and also residue monitoring system. This is very, very important, particularly for heavy metals, pesticide, veterinary drug residue, and mycotoxin. And veterinary drugs supply, sale, and use in animal feed. So that is also very, very important. And farming practice. So in large farm, there is no problem. We can ensure food biosecurity, vaccination, and management. It is more critical in small farm holding or small poultry farm. And European Union, they have regulation. They are saying that we should not use medically important antimicrobials for growth promotion purposes. And they put this issue in WTO SPS committee. So European Union, they are trying to minimize the use of antibiotics. And as our colleague from FSSI mentioned about Codex Standard Guideline and Code of Practice. So there are several documents. It is developed for rational, judicious use of antibiotics in animal production. As Madam has mentioned, we cannot think about poultry production without using antibiotics. So there are Codex Alimentarius Code of Practice on good animal farming, feeding, and Codex Alimentarius Commission guideline on application of risk assessment for feed, and revision of Code of Practice to minimize and contain antimicrobial resistance, particularly related to foodborne bacteria, Salmonella, E. coli, and so on. And there is also guideline on integrated monitoring and surveillance of foodborne antimicrobial resistance. So definitely, if we use in poultry or animals through food chain, we will get it. And that's why we have to match what is happening in human health sector and what new EMR gene are emerging in animal health sector that we have to match. There are international standard of feed and veterinary drug residue. So there is Codex Committee on residue of veterinary drugs in food. And for this Codex Committee, there is a JEFCA, Joint Expert Committee on Food Additives. They will take care of veterinary drug residue, veterinary drug residue contaminant in food. And based on that, there are YAMARIAL. And this committee, particularly this committee, which is hosted by United States of America, they determine priorities for the consideration, which type of antibiotics is widely used in animal health or animal feed, and recommend maximum level for such substances, and develop good practices if required, and method of sampling and analysis, how many samples should be taken, how to take samples so that it is acceptable for everyone. A total of 610 maximum residual limits for 75 veterinary drugs, including antibiotics. It has been developed so far, and WOHA set feed safety standard from animal health as well as animal welfare perspective. And WOHA also monitor antimicrobial use in food-producing animals and ban the use of medically important antibiotics in animal feed. So in that way, several organizations are involved in this fight against antimicrobial resistance. And this is the good practice of antimicrobial use in animal feed. It is from Holland. So we can see that it was used like turkey, poultry, pigs, and cattle. So there is dramatic reduction use of antibiotics in animal feed. So they improve hygienic, this biosecurity and vaccination. So it is doable. So that is the message I just want to give. And of course, we have to accept acceptable risk. We also take into consideration of consumer. So in Western world, consumer decide everything. So in Canada, I have taken this picture. So chicken that is raised without the use of antibiotics. Eggs from hens fed a vegetarian diet without animal byproducts. It is meat and bone meal. Because of that, there is Creutzfeldt-Jakob disease. And so in that way, these things are happening. And in the morning, we discuss about nutritional leveling. And also, we use minimum preservative or chemical in food, as little as possible. And there are acceptable risks, that is maximum limit, as well as maximum residual limit for toxin and chemicals, no use of growth promoter, hormone, and antibiotics, and attention to food allergy. So these are the trend of consumer conscious products. And there is also competition among multinational companies like McDonald's and others. Consumer concern cannot be ignored. So they are trying to reduce the use of antibiotics in animal production. So ideally is no antibiotics, no hormones. So that is the trend, and it is clearly shows that poultry and pigs, they are consuming a lot of antibiotics for supply of milk, meat, and egg to us. What is the way forward? So Basically here, this finally antibiotic bar on food producing animals. It was published in Times of India in 2012. So it is good that at least we came up with national standards. Enforcement is an important issue. So legal framework and its enforcement, that is very, very important. And code of conduct for judicious use of antimicrobials in animal feed, that is already there, and we need to work together. We cannot blame each other, but try to see how we can solve this problem. And there are also codex guidelines on application of risk assessment for feed, animal feed, and also integrated solvents EMO and EMR. So we have ESBL tricycle project, so that we consider E. coli as a universal microorganism that is found in human, animal, as well as food and environment. So we are trying to support countries to use this proxy indicator of contamination of environment with antibiotics. And risk mitigation measures like meal discard time. Yes, we have to, in mastitis, we have to use antibiotics. So if 3 to 5 days, if we do not use, then it will excrete it. So in that way, we can minimize the risk. And also withdrawal period. If we use antibiotics in animal feed for fattening animal, if we have withdrawal period, 1 month or 2 months, that will also solve a lot. And residue monitoring system, that is very, very important. And FSSAI, it has already started that kind of monitoring system. And that is important for consumer confidence. Last but not least, public-private partnership. Production is private sector, and quality control, quality assurance, that is the government sector or public sector. So there is FAO-led FAO and International Food Industry Federation. They develop manual of good practice for the feed industry for implementing the Codex Alimentarius code of practice on good animal feeding. So in that way, we all have to work together, and this is the global problem and global threat, and we all have to work together. Thank you.
Thank you, Dr. Gongal, for that precise presentation on antibiotic microbial resistance that may develop. But what I gather, or what I would like to sum up, is if you look at the animal production industry, they say that within 2030, the use of antibiotics in chicken will increase by 30%, whereas in the antimicrobial resistance, we are saying that we want to, by 2030, we want to reduce globally the use of antimicrobials for animals by 30%. This is a paradox, and I do not know how it is going to be achieved. Our next speaker in line is Mr. Srinivas Rao. He is the Head of Quality Assurance, Hatsun Agro Product Limited. I'm sure all of you are aware of Hatsun, and he will now give us his view on this subject and the challenges in the dairy industry. Mr. Srinivas Rao.
Thank you. It is a very simple. Milk is the only 4 letters it has, but it has so much of thing it is there. Our life will start with the milk, and again our day starts also with the milk only, with either tea or coffee. And in South India especially, without the curd rice, or without in the North India, without lassi or buttermilk, the day will not close. It is very, very important. It is a composite food. It will get all the fat, protein, carbohydrates, minerals, everything will get from the this milk. But we have so many challenges in the industry, where how it has to be taken up with respect to the, we are discussing only about the antibiotics only we are discussing, how we are going to see this one. These antibiotics, already we have discussed different antibiotics it is there. I want to see only some of the withdrawal time we have given this one here. Withdrawal time, each and every antibiotics has a different dates, times. That means if on antibiotic we have given 3 days, we have to wait. That means the body of the animal will metabolize the antibiotic, and it will give the milk of the free from antibiotic. That is, each and every antibiotic will have the different days. Okay, what is the problem with the antibiotic with respect to the human health? FSSI has given the so many 40 antibiotics has been given, the norms and MRLs has been given. But industry also is facing so much of thing if the antibiotic positive is there milk. We have given here this points it is there where that thing. Here this is the challenges it is there. Fermented product just like curd or yogurts and all, it will not set if a high levels of antibiotic because the microbial, the culture growth will not happen. Microbial culture growth will not happen. Microorganisms will not develop. And again the reduced yield. Yield also will not come, the cheese and all will not get the yield. Again with the high, okay, that is the risk of anti-resistance. Again the resistance is increased in the human body. Again it is because daily we are taking the milk. If it is more the antibiotics it is there, our body also will become the anti-resistance will happen. After that, whatever the injections we are taking antibiotic, it will not work. Today we have taken maybe 200 mg, next we have to give the 400 mg, next we have to go to 500 mg. After that, the particular antibiotic we have to avoid and we have to go for the next alternate antibiotic we have to go. This is a risk with this happen. And almost finally you see the highly stable in the pasteurization process. This whatever the antibiotics is there, we cannot do anything by there is no any system it is there, we cannot remove from the any step. Because it is from the animal it is coming, it has to be eradicated or controlled to the minimum level in the production in the animal area itself, we need to collect in the farm levels and all. What is the difference, why so much of problem we are facing in India compared with the Western countries? Western countries, they are having the farms, which is having 3,000, 4,000 cows, they will bring the milk, 10,000 liters they will supply in a day. There the control it is there, they will do the antibiotic testing and everything they will hand over to the dairy. But here it is not there, we are collecting the each and every village, 2 farmer or 3 farmer is supplying from the 4 liter to 10 liter they are supplying. It is very challenging to find out testing the antibiotics and rejection that level that is not possible into the ground level. This is the different type of antibiotics we are having. Different test methods also is available, which is a rapid test because milk is a very perishable product. Once it is come to the dairy, we cannot wait for the antibiotic results will come of the different, it may be 5 hours or 6 hours. We can't wait for the results because we need to safeguard the quality of the thing also. There are different types of test methods are there. And this is a microbial plating method it is there, and one is a spore-based Delvo and Eclipse methods are there. This will give the 3 hours, the results will come. And this is the ELISA kits are there, it is a fast, within 15 minutes we will get the results. And this HPLC and LMSS, this is a having, it will give the 1 day analysis time is required to get the quantify the results. And again, whatever the results it is there, again the factors also is getting, this is influenced by the test methods. Where the pH of the milk is there, if the high pH, that is where the milk is on the verge of spoil, it gives the false result. Maybe antibiotic negative, but it will show the antibiotic positive. And again, the presence of chemicals and preservatives. If any preservatives or chemical added to the milk, it will give the false results of the antibiotic positive, like that false positive results will give. And apart from the improper sampling, if the sample itself is not properly is collected, then again it will impact the totally testing and everything it will get impacted. And apart from the cross-contamination, mishandling of the milk, proper incubation time is required here. Maybe Delvo or Eclipse, it is there, 60 degrees, we need to incubate the milk. And here it is ELISA, it is 65 degrees, we need to incubate the milk. There, if the incubation temperature is not proper, it is plus or minus 5 degrees or something, everything, either positive may become negative, negative become a positive. That totally it will affect the, that means here the whatever the testing it is there, the accuracy is required, the people competency, the chemistry competency, the sampling and everything is very, very important to play into this one. This is a comparison with the testing to methods to testing to method testing. This is that here the accurate it is there, it is accurate. This is also accurate, it is high accurate, but it will take the time is consumers is a 1 day, 24 hours. We cannot wait for 24 hours, milk cannot be kept. And this is a beta star, it is there, we will find out in this method, the beta lactam groups we can quantify, verify positive or negative, but it will take only 50 minutes. But again, the cost is very, very high, that is also will come into the next stage, because unless the cost effectivity is not there, we cannot go to the intensive testing or farmer level testings and all is not possible this one. This is the 3 hours testing it is there, it will comes around 100 rupees or 120 rupees it is there. All these 2 is approved by the FSSI, all the 3 methods has been approved by the FSSI. This comes under the rapid methods, 15 minutes it is there. Both are the qualitative, this is the quantitative. It will give the 0.1, 0.2, 0.3 like that, it will give the quantitative. This is a cost comparison, it is there because it is so much of high is a cost with respect to 300 rupees and 3000 rupees, and again instrument cost is a lakhs of rupees need to be invested. In the field level it is difficult because as we told, the milk itself from the farmer level itself we need to control the microbials and everything. Otherwise in the industry there is no equipment or something to remove the antibiotics or like that, nothing is there. Only the option is available, negative milk and positive milk can make the blend and make it to the negative, like that the industry is practicing, is going on right now. This is the traceability again. From here we have shown the color here, green we have shown. In case of here the milk silo total overall we have collected maybe in the ABC, that means bulk coolers level BMCs. If we have collected 3,400, we can test this 4,000 liter. The totally traceability and backward and frontward integration both will come. We can identify up to this level, we can identify. But from here to the farmer, each and every farmer which is bringing the average maybe 4 to 5 liters of one farmer, it is very, very difficult to find out here the farmer level, which farmer has brought the antibiotic positive milk, how much it is there is very difficult. And apart from that, once we want to go back to the traceability back, by the 3 days, 4 days will be over, by withdrawal period also will be over. We are not able to track who is the farmer is bringing the antibiotic positive milk. But we are trying our level best to go back to the farmer level or village level testings also we are going. But that much of success we are not able to get. This is one of the automation it is there. Yes, we have comment like that one Eclipse method one it is given. Once the BMC tanker is loaded, they will check the antibiotic. They automatically, once they started the tanker loading is completed, they will put the LED. This is one of the monitor type it is there. Once the results will come, it will give the results will go to the cloud, and cloud will give the information to the dairy. By the tanker reach to the dairy, we will know that one is antibiotic positive or negative. By that we can take the decision, tanker can be unloaded or we have to wait for some time to confirmation tests and all like that. Recently we have started this in our Hudson plants. In the BMC level we have started this one. By that once a tanker coming, we will know that one this is a positive or negative. Only recently we have started some 20 or 30 areas we have started this one. This is the conclusions. Only we need to give the farmers education. Right now there is no farmer, legally nothing is there about the farmer. We need to educate the farmers why the antibiotics, what is the levels of use. We cannot avoid the usage of antibiotic because animal is important, animal health. In case of any animal health is problem is there because lakhs of rupees cattle value. They need to give the administration, but they should know how many days the milk should not be given. That all the awareness we need to give to the farmers. And supervision at the farm levels, where the farm level, that is only again here also the farm level means what we want to say, we are supplying the cattle feed from our own cattle feed manufacturing units are there. From there we will give to the farms by the good quality of feed can be given, by that animal health will be taken care. That will taken care only, it is exclusively supplied to our farmers only will supply. And cost-effective antibiotic testing, where the now 60 rupees, 100 rupees or 90 rupees, 300 rupees is there. It is a cost-effective, further more cost-effective antibiotic testing solutions are required. Then only we can go intensive testing to the farmer level, we can take up, we can educate the farmers. Because without knowing which farmer is antibiotic positive, we cannot train them, they will not accept also. That type of issues also is there. And testing and quality assurance programs, what the people who is doing the analysis part and all, we need to, so much of testing and intensive programs, trainings and all we need to given. Then only we can assure the antibiotic negative milk can be given to the market like that. But even though we are in the number 1 in the world milk supply, tomorrow it is exports will be very, very difficult to export the milk and milk products. Even though FSSI has given only 3 months once testing. But as at our factory, we will do each and every tanker will be tested for the antibiotics. After the antibiotic clearance only, the product will be given to the market supply will do at our Hudson plants. That is the thing. Thank you.
Thank you, Mr. Srinivasan, for that overview. I do agree with you that, responsible sourcing of the material is most important for the food business operator to do that. But in terms of rapid kits, I would like to tell you, and the industry who are involved in dairy and dairy products, we have in FSSAI got approval kits, RAFT approval, that is the rapid testing kits approval there. And there we take care when any kit is approved for the testing, we look at the validation, and the validation has to be done by a third party, generally by Microval, AFNOR, AOAC or thing. And there what you suggested, that the pH of the milk or the fat content, there all these are studied. In fact, when we do a validation, we do what is known as ruggedness, especially for milk. We do it with high fat, high pH, low pH, high somatic cell count and things. So I would advise the industry, if those of you who want to use these kits, which I think we have for colistin and something, we have rapid kits. I'm not sure because I'm the chair of the committee and we have approved some of the kits, but the list is available in the FSSAI website on which kits have been approved by FSSAI. But yes, I agree, in India and the third world countries or developing countries, most of our farmers are, small hold, 2 cows, 3 cows, that's their daily livelihood or thing. Unlike in the West, where they have a ranch which is like thousands of acres and they have, cattle as well as the dairy cows or thing. This is a problem. And the third thing that you talked about is the awareness, and this morning we have been hearing about the culture. I think that culture has to be developed, because what has happened among our farmers and things is they are not aware, they are not educated, and so they think, giving antibiotic till 8 days is a long time for them for withdrawal period, because they lose 8 days of earning. So they tend to, supply the milk earlier, which is there. But it's heartening to note that you are, having that system which uses the biosensors, and you're able to, tell the milk that is there. But what I would suggest is maybe, you should get that method validated and see whether really, what we're talking about is quality assurance. And so, in any method that is used, it must be a validated method, which takes care of accuracy, precision, ruggedness, robustness, and everything. Thank you very much for enlightening us on, what the dairy industry faces due to the antibiotics. May I now request Dr. Edward Danish from Fresh to Home Foods and Private Limited, the Chief R&D Officer, to give his views on this subject. Mr. Danish. Thank you.
Good evening, and I would like to thank Food Safety Works for giving me this opportunity to talk about our operations. So regarding antimicrobial residue resistance, the speakers before me have given a very good overview of the topic. So I will restrict myself in that we are also running out of time. So to just give an overview of what we are doing as a what you call procurement organization, and an organization which sells fish, meat and chicken to the customers. Now for us, as we front end and we reach and we talk and we interact with the customers, the customer requirement becomes a very important thing. And as you all know today, customers have lot of what you call awareness about antibiotics, about hormones, about different kinds of chemical residues. They will go and Google and they will read and they will call us back, and they will call our customer service officers and they will eat their heads. So we have prepared a lot of FAQs, and we will, whenever they can't handle, they put them on the call to me. And then I will explain to them what we are doing. And sometimes even my own families and relatives, they will ask me so many questions. So many times I tell them, you know, you are most welcome, you are invited, anytime you please let us know and come, we will take and show you our operations. So that is the way we are working with our customers. So basically, the speakers before me have already described how the antibiotic use in livestock is leading to resistance, and then we know that the customers, human beings, when they get exposed to this kind of meat or this kind of products, they will whenever they suffer instance of infection, the antibiotic will not work on them, and it is a very what you call difficult thing, because whatever medicine is there which is available, and recently one of my friend did suffer this issue, and the treatment given for typhoid was not working on him, and he was admitted in the hospital for a very long time. I have personally come across such conditions. So not only not only use when the animals are sick, there is also a prophylactic use that the farmers do when the animals are also healthy. You know, there will be a seasonal change. They will expect some kind of infection, so they will. It is like in Tamil they will tell "Thalaiya sutti podradhu." So they will just treat the animal with some kind of medicine, and they believe that that particular problem will not come, but they are not understanding that they are compromising the health of their animal and also the byproduct, the health of the human beings who will eat that byproduct. So it is a very common issue in animal production. So how we have solved it, solve this problem is what I want to focus on here. So what we do is for our poultry production, we have a pre-approval process. So we have a veterinary team led by a veterinary doctor, and he will do the integration part, and we have a nutritionist and feed designer who will design the feed, and we make our own feeds for our for our chicken farmers. So what the veterinary team will do is they will go to the field and they have a interaction with the farmer who want to give their farms for the integration. So they will make an assessment and there is a checklist. So the whole facility will be inspected. So we have lot of focus on biosecurity. So the entry of pathogens, or whether the cultured birds or whether the birds are under during the growing period, if it is going to interact with any wild birds. So the pathogen entry points, the interaction of the people, how the feed is stored, delivered, all these things we will do a risk analysis and we list it out, and then we have a very strict control, and we are systematically giving the feed as per requirement to the farmer so that the dispersion and cross contamination does not happen. And also the stage wise, the kind of protection that the chicken is requiring at each stage, what kind of barriers are required, they are assessing that, and they are implementing those things in the farm. And then whenever in some case there is some instance where some incident has happened and some application is there, then they will make sure regarding the enforcing the withdrawal period. And then if there is any concern, they are now again taking the animal and they will get it tested, and then they will decide whether to continue with that batch or not, or whether to destroy that batch, and then they will go to the next batch. So that is the next level. And also they train the farmers, and also we train our field staff for doing this kind of observation, and proper record keeping is there, and all this data for each farm, each batch, we are maintaining a health record for the number of days of culture for whatever period the animal is there. And so when we get a batch of animal for processing, we know from which farm, which set of chicken has gone to the process plant, under what this thing, and the veterinary inspection records, the AM/PM records, and the process batch number will be there. And when it receives to our factory, we can, the whole thing is put into a computer, and we have an internal software where we put a batch number, we can get the full backward traceability right up to from which farm the bird has come. To this extent, we can trace in this thing, and the same we have implemented in certain fishes in the aquaculture farms also for fishes. So this is the level of traceability that we are able to implement in the poultry end. And meat, we like the goat and sheep meat, we do not purchase from smaller farmers. Well, we are getting it from export houses. So thereby all the traceability documents and testing is completed. So already I have highlighted about the awareness and market pressure which the customers are already having. Most of the customers who buy online from us, they are very well educated and they do have a lot of knowledge. So we try to, whenever they ask us anything, we try to explain the issues to them. So to summarize, so we work a lot on biosecurity to prevent the entry of pathogens, and wherever treatment is done to make sure that the proper withdrawal records are there, withdrawal period is maintained, and the records for such withdrawal periods are maintained and available. We also give a lot of training to the farmers on good farming practices, and we are designing our own nutrition and supplying to the farmers the hatchlings, the fish seeds and the feed, all these things we are delivering to the farmers. So this is a very important concept of partnership that we are able to develop with the farmer, and we are able to because of that we have a lot of interaction, and with a lot of confidence we are able to place this product that we have with our customers, and whatever questions they have we are able to answer them. And with regards to testing, we follow a very strict testing calendar we make, and all the regulatory requirements and all the our internal requirements, monthly testing, and we also publish these reports on our websites for customers' benefit. So these reports we will be sending it to NABL accredited labs, and through them we get these tested, and whatever tests are there we are publishing them. So this is how we are implemented the requirements, mandatory requirements, our customer requirements, and how we can give residue-free, a clean label product to our customers. Yeah, that's the end of it. So if any questions, we could take it. Thank you.
Thank you, Dr. Edward, for that very short and precise presentation that you had. And I think what we have seen in the last two presentations is a difference, at two ends of the spectrum, where Hatson is, sourcing from all farmers, whereas Fresh to Home has got their own, secluded farming, or what I would say is contract farming. So they are able to control the whether it is the antibiotic residues or pesticide residues, whatever is being used. And they also see that their feed is manufactured by themselves. So there is a large difference between, sourcing globally from other things and this. So whereas they also have, although they do this, their own contract farming or thing, they have very well in place things for their testing pre-approval. And it's heartening to note that you're very transparent, and we can look at your reports if we go to the website, because I think that is what actually increases the consumer confidence on the product, when we are able to look at test reports. Not everyone would look at it, but that is there. I will have to end this session for paucity of time, and maybe at tea time you could have this, because Dr. Gongal has to leave. He has a flight at 8:30, so I will have to wind up and hand over the things. So maybe at tea time you can network with Mr. Srinivasan and Dr. Edward on the thing. And I would thank all the three speakers for their excellent presentations and keeping the time. I think our session made up for the lost time of the previous sessions. So thank you, Dr. Gongal, thank you, Mr. Srinivas, and thank you, Dr. Edward, for this. And I thank the audience for your patient hearing. Thank you.
Just I want to say here that the key message is if animal feed is not safe, we are not safe. That is the main thing.
Thank you so much, sir, for the thoughtful words. I'd like to request Dr. Lalita R. Gowda to honor all our panelist members. Sir, please come to the stage. Thank you. Dr. Gyanendra Gongal. Thank you, sir. Can you come to the center, sir? Can you, yes, to the center. Yes. Yes. Give them the bag. Thank you so much, sir. Thank you, Mr. Srinivas Rao, sir. Wait. Give the kit. Thank you. Thank you, Dr. Edward, sir. I'd like to request everyone to the stage for a photo. Thank you all for the intriguing session. I hope this discussion was insightful.